All right, we're going to go ahead and continue with 15.2. Hopefully you enjoyed Bosman. I think he does a real good job. And kind of enjoyed, I know it was a little bit of a review in the beginning there when he talked about the bird beaks, but I thought it was neat how he took and used that and showed y'all application into chapter 15. All right, let's go ahead and get on through for your notes. The change of population allele frequencies. Remember Gregor Mendel with his famous pea plants. He was not respected when that originally happened. Remember, all of Darwin's stuff was coming about the same time, and it wasn't until the 1900s, the early 1900s, that biologists began to take a look at his work again. And they wanted to study how the frequencies of alleles changed in a population. So let's look at that terminology, frequencies of alleles. In other words, remember what those alleles are. We use them to recognize, when we look at homozygous dominant, big A, homozygous recessive, little a, those represent the alleles. If we have a gene for whether or not you have hitchhiker's thumb or not, whether you do have it or you don't have it, those are the alleles for that gene. All right, so in 1908, the English mathematician G.H. Hardy and the German physician Wilhelm Weinberg independently demonstrated that dominant alleles do not automatically replace recessive alleles. Their discovery, called the Hardy-Weinberg principle, states that the frequencies of alleles in a population do not change unless evolutionary forces act on the population. All right, so this holds true for any population as long as that population is large enough that its members are not likely to mate with relatives, and as long as evolutionary forces are not acting. Here are the 5 major principles: mutation, gene flow, non-random mating, genetic drift, and natural selection. So these cause those small evolutionary changes to occur. These are considered evolutionary forces. So let's look at mutation first. Although mutation from one allele to another can eventually change allele frequency, mutation rates in nature are very slow. Furthermore, not all mutations result in phenotypic changes. And let's think about what phenotypic is, what the phenotype is. It's what we physically see. So if we have this mutation in DNA, it is not necessarily going to result in the RNA being mutated, or if the RNA is mutated and it's not caught, it doesn't necessarily mean that when you start stringing together those amino acids that it's going to make a huge difference in what that protein is. Remember when we did transcription and we were coding for those amino acids? Several different 3-sequence codons, the 3 nucleotides, coded for one specific amino acid. So sometimes those mutations aren't going to change what that code would be. Mutation, however, is a source of variation, and thus does make evolution possible. I'm going to skip that one. Let's talk about gene flow. Gene flow, the movement of individuals from one population to another, can cause genetic change. So we're talking about whenever individuals move in or move out. So their alleles move in or move out. So they're flowing. Those genes are flowing. Gene flow occurs because new individuals, and I like to say immigrants, add alleles to the population. And if you have departing individuals, emigrants, like exit, they take these alleles away. Oh, sorry about that. All right, so we talked about immigrants, immigration, This is where you have new alleles come into the population. Or if you have alleles taken away, that means individuals are departing, exiting, immigrants. I do want to show you this.
Flowers in this valley are the same species. The gene that codes for flower color has two alleles, one for red color and another for purple color. On one side of the valley, purple flowers flourish, on the other, red flowers flourish. Normally, the wind blows in the direction that the water flows. This creates a natural boundary between the populations. Pollen from the red flowers is not able to get to the purple flowers to pollinate them. However, a temporary change in wind direction causes pollen from the red flowers to blow across the river, allowing the pollens with alleles for red color to move to the hillside with the purple flowers. By chance, the pollen grain carrying the allele for red flower fertilizes a purple flower. After a couple of generations, there are now a few red flowers on the purple flowered hillside. Some red alleles have moved or immigrated to the purple hillside. The migration of the pollen functions much like the movement of individual organisms into or out of a population. The process of alleles moving from one population to another is called gene flow.
Okay, moving on. So now let's talk about non-random mating. And I think this is always a fun one to talk about with y'all. I wish I were there. Sometimes individuals prefer to mate with others that live nearby or are of their own phenotype. This is a situation called non-random mating. Now, usually in humans, we have non-random mating. Normally, people are pretty particular about who they decide they want to mate with. Well, that depends upon what time they get out of the bar, I guess. No, just kidding. All right, mating with relatives or inbreeding is a type of non-random mating that causes a lower frequency of heterozygotes than would be predicted by Hardy-Weinberg principle. So if you don't have this, if you don't have random mating and you choose to mate with your relatives, remember we're not always talking about humans here, then that's going to cause a lower frequency. That is one of the reasons that humans do not normally inbreed, is because if there are problems in their DNA, if there are genetic defects in their DNA, any of those recessive diseases or traits will come out much more often if they inbreed. Non-random mating also results when organisms choose their mates based on certain traits, and I have some real neat videos to show you on that, but we won't do that today. We're going to go ahead and move on. Let's talk about genetic drift. Think about this, genetic drift. All right, so listen to the terminology to help you remember it. In small populations, the frequency of an allele can be greatly changed by a chance event. So we're talking about small populations here. Because this sort of change in allele frequency appears to occur randomly, as if the frequency were drifting, it's called genetic drift. Okay? So if an allele, this allele frequency occurs randomly in these small populations, it appears as if it were drifting because it happens randomly. It's called genetic drift. Small populations that are isolated from one another can differ greatly because of this genetic drift.
In small populations, the frequency of an allele can be greatly changed by a chance event. The failure of even a single organism to reproduce could significantly disrupt the allele frequency of a small population, or a predator might kill the only member of a population with an unusual allele. A particular allele may disappear completely over a few generations. In a larger population, a particular allele may vary widely in frequency due to chance, but still be present in enough individuals to be maintained in the population. The death of a few organisms or failure to reproduce among some organisms may alter the allele frequency in a few generations, but will not result in the extinction of the allele. In a much larger population, the frequency of a particular allele may vary slightly due to chance, but will remain relatively stable over generations. Because a large number of individuals carry the allele, the overall allele frequency is not significantly affected by the death or failure to reproduce of a few individuals. The blue line shows how the allele frequencies in very large populations are generally very stable over time.
You see the importance. It's So you see the importance here of a large population and the difference it can make. All right, so natural selection, this should be a good review. Natural selection does cause deviations from the Hardy-Weinberg proportions by directly changing the frequency of alleles. The frequency of an allele, how often these alleles occur, will increase or decrease depending on the alleles' effects on survival and reproduction. Remember, that's what natural selection is. It is basically fittest of the survivors. Those that are best suited to the environment are going to be able to reproduce, thus passing on their alleles. We're going to go ahead and move on to that. How natural selection acts. Only characteristics that are expressed can be targets of natural selection. Therefore, selections cannot operate against rare recessive alleles, even if they are unfavorable, because they happen so infrequently. Only when the allele becomes common enough that heterozygous individuals come together and produce homozygous, normally homozygous recessive offspring, does natural selection have an opportunity to act. Many human diseases caused by recessive alleles have low frequencies. Genetic conditions are not eliminated. I'm sorry, I can't talk. Genetic conditions are not eliminated by natural selection because very few of the individuals bearing the alleles express the recessive phenotype. Just think about that as homozygous recessive. Those are much less common. Natural selection shapes populations affected by phenotypes that are controlled by one or by a large number of genes. A trait that's influenced by several genes is called a polygenic trait, which we've talked about that before. Polygenic traits tend to exhibit a range of phenotypes clustered around an average value. If you were to plot the height of everyone in our class on a graph, the values would probably form a hill-shaped curve. That's called normal distribution. So we'd have a few short people and a few really tall people, and then we'd have a wide range of those that are kind of in between everybody. This hill-shaped curve represents what's called a normal distribution. This is what normally would happen. The blue dashed line represents the average height for this population. Now, we can have directional selection. This is one of the things that Bosman talked about. When selection eliminates one extreme from a range of phenotypes, the alleles promoting this extreme become less common in the population. Well, that kind of makes sense. If you're not around to pass on your genes, if you've been eliminated, if you're real short and you've been eliminated because you couldn't reach enough food or you weren't able to be big enough to fight off whatever, then you're not going to be able to pass on your genes. So therefore, those genes that carry the shortness won't be wiped out completely, but they will be much less common. In directional selection, the frequency of a particular trait usually will move in one direction. I'm going to move on to that. We will watch this one. I like this one.
Consider a hypothetical species of anteater. The blue curve on the graph shows the tongue length of a population of anteaters.
Short,
tongue length
long.
increases to the right. You can see that most anteaters have a medium-length tongue, with a few anteaters having a very short or a very long tongue. Anteaters feed by breaking open termite nests, extending their tongue into the nest and lapping up termites. Suppose the area in which these anteaters live is invaded by a new species of termite that build very deep nests. Anteaters with long tongues will be able to more effectively prey on termites deeper in the mound than can anteaters with short or average tongues. Thus, anteaters with short tongues may not survive to reproduce. Directional selection would select against shorter tongues for anteaters. The red curve shows the variation in tongue size several generations after this new species of termite was introduced. Notice that there are not as many medium tongue length anteaters. There are far fewer short-tongued anteaters and many more long-tongued anteaters.
which Bosman used the example of the beak size, which I thought was really good. All right, stabilizing selection. When selection reduces extremes, whether like with the last one we just saw, the very short tongues or the very long tongues, in the range of phenotypes, what we physically see, the frequencies of those intermediate phenotypes are going to increase. As a result, the population contains fewer individuals that have alleles promoting extreme types. So you would have less long-tongued and less short-tongued anteaters. In stabilizing selection, the distribution becomes narrower, tending to stabilize the average by increasing the proportion of similar individuals. And take a look at that.
Consider a hypothetical species of lizard. The blue curve on the graph shows the initial variation in lizard size as a standard bell curve. Body size increases to the right. The dashed line indicates average body size. In stabilizing selection, extreme body types are selected against. For example, consider a hypothetical species of lizard in which predators eat the smaller lizards which cannot run fast enough to escape. Predators also eat the larger lizards, which are more easily spotted, captured, and eaten. So the larger lizards and the smaller lizards do not survive to reproduce. The red curve represents the variation in body size several generations after the new predator, which easily captures large and small lizards, is introduced. Notice that there is still the same number of medium-sized lizards, but there are now far fewer large and small lizards.
All right, so this gives you a good picture of what directional selection is, how we both start out. This is our average value. In directional selection, it will move one direction or the other to those extremes. In stabilizing selection, though, it will narrow down, and those extremes will disappear. All right, we are done with that. All right, I want to show you one just a little bitty short, less than a minute video. And when you're done with it, before you start your worksheet, in your bell ringer today, since we didn't do a bell ringer, put today's date down, April 5th, and I want you to draw a graph showing directional selection with these moths. Here we go.
Before 1850, the peppered moths most often captured by collectors were light in color. Their pale gray coloration resembled the lichen-covered tree trunks on which they rested during the day. In the 1850s, the English Industrial Revolution got underway, and sooty smoke began to blanket the countryside. Soot and other pollutants killed lichens and darkened tree trunks. Collectors found that dark-colored moths were now captured more frequently. The collectors hypothesized that conditions had previously favored light moths, but now favored the dark ones. Directional selection had shifted the range of variation for moth wing color.
All right, so I want you in your bell ringers to draw two graphs, one showing what the population with dark and light moths were initially. And with the populations, you know, the collectors caught many more white ones than they did brown ones. Then once the Industrial Revolution occurred, they captured many more dark-colored ones. And so if you will draw both of those graphs, I want you to try to figure out what would be on the X and the Y axis. Think about what is the dependent and the independent variable. What depended upon the other. All right, now you can go ahead and get to work. Once you're done with that, you can go ahead and get to work on your worksheet. It is due on Monday. Now have a great weekend.